Hello students, I'm going to discuss the quick solutions of All India AIATS Test Series Medical 2020 of test number 8 code C and D. Your test was held on 31st of March 2019 and I'll be dealing with your botany section. The very first question in the botany section is question number 121. How many of the following are defining features of living organisms? First is metabolism, responsiveness to external stimuli, reproduction, growth by accumulation of material on surface, cellular organization. First option 5, second 4, third 3, fourth 2. In this particular question, you have to tell that how many of the given features are defining features. The features to be defining should follow some criteria such as these defining features should present in all the living organisms without any exception, and the second is the defining features should not be shown by any known living object. Now see one by one. Metabolism, yes. Metabolism is shown by all living organisms, whether they are unicellular, multicellular, prokaryotic, eukaryotic, any kind of organisms. So it's a defining feature and it does not shown by any kind of non-living object. Responsiveness to external stimuli, yes, it is consciousness, and consciousness is also the defining feature of all living organisms. Reproduction, no. Reproduction is shown by living organisms only, but there are some exceptions in the reproduction. For example, mule, worker honeybees and infertile human couples, they cannot reproduce at all. So reproduction cannot be a defining feature. Growth by accumulation of materials on surface, this is not the defining feature. Why? Because it is shown by the non-living objects. Living organisms show intrinsic growth, means from inside the body, which results in the increase in the number of cells and the mass. But here, in non-living objects, there is an accumulation of materials on the surface. For example, sand dunes, mountains, they show extrinsic growth. So this is not the defining feature. Cellular organization, yes, it is also a defining feature because it is shown by living organisms only and without exception. So 3 are the defining features. So the correct option for the given question is option number 3. Let's move to the next question. Question number 122. Select the incorrect statement. First, plant cell wall is chiefly composed of cellulose. Second, in ripened fruits, middle lamella, which is made up of calcium and magnesium pectate, remains intact. Third, plant cell wall and middle lamella may be traversed by plasmodesmata, which connect cytoplasm of neighboring plant cells. Fourth, in mature plant cells, secondary cell wall is added internal to the primary cell wall. You have to identify the incorrect statement which is mentioned here. See one by one. Fourth one is, in mature plant cells, the secondary wall is added internal to the primary cell wall. Yes, in plant cells, when secondary cell wall deposition takes place, it is always inside the primary cell wall. So this is correct. Third one, the plant cell wall and the middle lamella can be traversed by plasmodesmata. Yes, and this plasmodesmata connects the cytoplasm of 2 neighboring plant cells. Now the first one, the plant cell wall is chiefly composed of cellulose. Yes, plant cell wall is cellulosic in nature. It is also correct. See the second one, that in ripened fruits, middle lamella, which is called the cementing material, it is made up of calcium and magnesium pectate. True, but it does not remain intact in the ripened fruits. It gets solubilizes and makes the fruit soft. So this is the incorrect statement. So the correct option for the given question is option number 2. Let's move to the next question. Question number 123. Match the given columns and select the correct option. In column 1, there are substages of the prophase 1, which is a stage of meiosis 1. And in the column 2, there are some events which take place in these stages. So you have to match the items in the column 1 to the items in the column 2. A. Diplotin, terminalization of chiasmata. B. Pachytene, dissolution of synaptonymal complex. C. Zygotine, formation of recombination nodule. D. Dikinesis, formation of bivalence. Now see, in case of meiosis 1, there are substages called prophase 1, metaphase 1, anaphase 1 and the telophase 1. This prophase 1 is subdivided into many phases which are called leptotene, zygotene, pachytene, diplotene, and dikinesis. So what happens in these stages? In leptotene, there is condensation of the chromatin material, but in your question, leptotene is not mentioned. Second is zygotene, in which there is pairing of the homologous chromosomes called synapses, which results in the formation of bivalence. So C matches with fourth. Option number 1, 3 and 2 are ruled out, as in only a single option C matches with the fourth. So this is correct. Next is pachytene. In case of pachytene, there is crossing over takes place, and to take place the crossing over, there is formation of a combination nodule. So B matches with the third. It is also correct. Now diplotene, what happens in the diplotene? In case of diplotene, there is a dissolution of the synaptonymal complex. What is synaptonymal complex? This complex ensures the togetherness of the two homologous chromosomes so that crossing over easily takes place. So A matches with the second. And the last one is dikinesis, in which terminalization of chiasmata takes place. So D matches with the first. What is chiasmata? It is the X-shaped structure which is seen when these two homologous chromosomes after crossing over separate but still united at a point which is X-shaped and called the chiasmata. So this terminalization of chiasmata takes place in the dikinesis. So the correct option for the given question is option number 4. Let's move to the next question. Question number 124. Two pathogens A and B can pass through bacterial filters, but B can only multiply inside a living host, while A can grow on culture medium as well. Select the option that correctly identifies A and B. First option, cyanobacteria actinomycetes. Second, bacteriophage prions. Third, mycoplasma viruses. Fourth, prions virusoids. So two pathogens A and B can pass through bacterial filters. It means both the pathogens are smaller than bacteria. And B can only multiply inside a living host. It means B is some intracellular parasite, and because it can only multiply in the living host, it is obligate intracellular parasite. And while A can grow in the culture medium. What should be the B? Actinomycetes? No, they can grow outside also. Prions, viruses or virusoids. Yes, it can be any option. Now, A can grow on cultured medium as well. It means it should be smaller than bacteria and should grow on cultured medium. It should be mycoplasma. So the correct option for the given question is option number 3. Let's move to the next question. Question number 125. A feature which is not common between sac fungi and club fungi is: first option, presence of septate and branched mycelium. Second, formation of fruiting bodies. Third, presence of dikaryophase in their life cycle. Fourth is presence of sex organs. What are sac fungi? Sac fungi are ascomycetes. And what about the club fungi? They are basidiomycetes. So the feature which is not common about the ascomycetes and the basidiomycetes is presence of septate and branched mycelium. No, it is present in both of them. Formation of fruiting bodies, it is shown by both of them. Presence of dikaryophase, it is also shown by both of them. Although the dikaryophase is shorter in the sac fungi as compared to the club fungi. Presence of sex organs, yes, sex organs are only present in the sac fungi but absent in club fungi. In club fungi, plasmogamy takes place by somatogamy. So the correct option for the given question is option number 4. Let's move to the next question.